Hello everyone, my name is Favian Reswara. I'm coming from Biotechnology major in Universitas Unggul. In this video, we'll discuss how algae from microscopic phytoplankton to giant seaweeds capture carbon and how industrial application can use those processes to reduce emissions and how modern genome editing technologies like CRISPR can accelerate algae's capacity to act as more efficient carbon factories. Algae are responsible for a large share of global primary production. Marine microalgae alone fix millions of tons of carbons each year, and they drive the biological pump that moves carbon from the atmosphere and the ocean surface into food webs and sediments. Because of their fast growth rates and photosynthetic efficiency, algae are natural candidates for biology-based mitigation strategies. At the basic level, algae use the Calvin cycle to reduce carbon dioxide into sugars. The key enzyme is rubisco, but rubisco has a weakness. It can also react with oxygen, causing photorespiration and carbon loss. In aquatic environments that dissolve carbon dioxide, the availability is often low, so many algae have evolved carbon concentrating mechanism, or CCMS, to raise local carbon dioxide concentration around rubisco. CCMS operate via ion pumps or transporter that takes up bicarbonate, the enzyme carbonic anhydrase that rapidly converts bicarbonate into carbon dioxide, and specialized organelle structures like phyronoids in eukaryotes or carboxysomes in cyanobacteria that cluster rubisco and retain carbon dioxide so it doesn't leak away. CCMS are a major reason algae can maintain a high photosynthesis even at low ambient carbon dioxide. The phyronoid is a phase-separated organelle found in many eukaryotic algae. It concentrates rubisco together with matrix or scaffold protein so that carbon dioxide remains high in the reaction zone. Structural studies and modeling show that reducing carbon dioxide leakage around the phyronoid and arranging enzymes correctly are key to an efficient CCM. In cyanobacteria, the carboxysome plays an analogous role, enclosing rubisco in a protein shell to trap carbon dioxide. Newton experiments and modeling studies have helped formulate the biochemical and physical principle underlying efficient CCMS. We should distinguish two major groups of algae. First, microalgae, such as chlorella, diatoms, nanochlorophysis, all of them are single cells, and macroalgae, like seaweeds, such as kelp. Microalgae excel in productivity per unit area and suit closed photobioreactors or raise low costs. They can be stirred to produce lipids for biofuels, carbohydrates for fermentation to bioethanol, or bioplastic precursor polymers. Microalgae grow large in open oceans without needing arable land or fresh water and are suitable for large-scale scenarios such as ocean farming that exports biomass. The choice between them depends on goals. High-value biofuel and pharmaceutical products tend to use controlled microalgae systems. Large-scale mitigation and biomass sinking strategies point towards microalgae. There are three main pathways commonly discussed in tangled industrial application to reduce emission by using algae. Firstly, algae biofuels. Microalgae lipids can be converted into biodiesel if production is supplied with industrial carbon dioxide and biomass is rotated back into the system. The cycle can be carbon neutral. Coupling production with carbonate storage could yield real emission reduction. Secondly, bioplastic and value-added materials. Algae can be feedstock for PHAS, PLA precursor, and other polymer plastics. The final products will carbon into biodegradable materials if managed properly. Thirdly, or finally, biological CCUS, or algal-based CCUS. Placing algal cultivation units close to emission source, like power plants or factories, allow them to absorb waste of carbon dioxide. Pilot study shows such system can work on a limited scale, though challenges remain in scalability, energy needs, and infrastructure costs. In short, algae breaks carbon dioxide uptake and products that can replace fossil-based materials, but every pathway needs rigorous life cycle assessment to ensure actual emission reductions. Although algae are highly productive under lab conditions, real-world deployment face problems. Environmental fluctuations such as temperature, light, and salinity, predators and contaminants in open ponds, carbon loss, fire respiration, and remineralization and metabolic trade-off. Additionally, harvesting and drying biomass remain energy-intensive and costly. Therefore, we need improved algal strains that are stable and directed to produce desired products while keeping high carbon fixation rates. This is where CRISPR comes in. In recent years, CRISPR-Cas editing tools have become useful in model microalgae, such as Chlamydomonas and nanochlorosis, and some cyanobacteria. Common strategies to improve algae related into carbon cycle metabolism can be included by, firstly, redirecting carbon flux, knocking out storage pathways such as synthesizing enzymes and activating lipid or PHA pathways to increase the share of biomass turned into valuable products. Secondly, strengthening CCMS by introducing or enhancing genes for bicarbonate transporter, carbonic anhydrases, or pheromone structural factors so algae better capture and retain carbon dioxide. This can reduce the need for external carbon dioxide input and raise productivity. Thirdly, improving environmental tolerance by the algae, overexpressing antioxidant enzyme, heat shocks, chaperones, or assimilate pathways so strains survive fluctuating outdoors hunt that lower crash rates and improve animal productivity. Finally, by improving or editing the rubisco protein. This remains technically challenging, but approaches like base editing or prime editing aim to improve rubisco kinetics or regulation so carboxylation is vapor over oxygenation. This is an active research area. So some studies have shown early success by higher lipid accumulation after disrupting storage genes, improved PHA production, and enhanced stress resilience, though many results are at laboratory scale and require validation at the field scale. Imagine an ideal strain for CCUS and biofuel. Firstly, a more active biocarbonate transporter. Second, a stable pheromone that prevents carbon dioxide leakage. Thirdly, lipid pathway that active under nitrogen limitation. And finally, stress regulators that preserve photosynthesis at high temperatures, all controlled by CRISPR-A or CRISPR-I circuits and permanent edits. In the lab, combining strategies like this has improved certain performance metrics. The challenge is maintaining those gains in the open ponds throughout a full year.
We must acknowledge risk. Releasing engineered algae into open waters raises ecological concerns about gene flow and invasiveness. There are socio-economic questions: who benefit from algae carbon farms, and how are coastal or freshwater rights managed? Responsible developments require robust containment strategies, environmental risk assessments, transversal stakeholder engagement, and regulatory pathways that balance innovation with safety. Research should include ecological trials and modeling before large-scale deployment. In conclusion, algae have great capacity to fix carbon and serve as circular feedstock for for fuels and material. Technologies such as CRISPR give us tools to accelerate and direct that potential, but they must be combined with engineered systems like reactors, harvesting, and logistics, and cautious policy. If developed responsibly, engineered algae could be a meaningful component of a broader climate mitigation portfolio that blends the natural approach with advanced technologies.